Welcome and greeting to all. My name is Dr. Irfan Ahmed, working as a medical lecturer in Samarkand State Medical University, Department of Microbiology, Virology and Immunology. So today our topic is physiology of bacteria, growth and reproduction of the bacteria. Lecture schedule: the chemical composition of the bacteria, cell metabolism, constructive metabolism, types of microbial nutrition, bacterial transport system, types of respiration, growth and reproduction of the microbe, nutrient media. The metabolism refer to all the biochemical reactions that occur in a cell or organism. The study of the bacterial metabolism focus on the chemical diversity of the substrate, oxidation and dissimilation reaction by the which substrate molecule are broken down, which normally function in bacteria to generate the energy. The chemical composition of the bacteria is: protein is 55%, total RNA is 20.5%, DNA is 3.1%, phospholipid is 9.1%, lipopolysaccharide is 3.4%, and murine is 2.5%, and inorganic ions is 1.0%. So next move forward with the heading of the bacterial cell consists of water 70 to 90%, dry weight is 10 to 30%, protein 55%, 2.35 millions of the molecules, 1850 different type of molecules, RNA 20.5%, 25,000 molecules, 606 different type of the molecules, DNA 3.1%, 2 molecules, lipid 9%, 22 million of the molecules, lipopolysaccharide 3.4%, 1.5 million molecules, peptidoglycan 1 molecule. So microbial metabolism consists of the catabolism and anabolism. Catabolism is a dissimilation pathway that break down the organic substrate, means carbohydrate, lipids and proteins, to yield metabolic energy for the growth and maintenance. The anabolism is assimilation, assimilatory pathway for the formation of the key, intermediate and then to end product cellular components. Intermediary metabolism integrate to process. You can see the catabolism, substrate level, the phosphorylation, fermentation and aerobic respiration. The bacterial cell is a highly specialized energy transformer. The chemical energy generated by the substrate oxidation is conserved by the formation of the high energy compound such as adenosine diphosphate and adenosine triphosphate are compounds containing the thioester bond. Another form of energy is a transmembrane potential, chemiosmosis, production of the ATP in electron transport. The electrochemical gradient formed between the membrane, hydrogen proton generated from the NADH electron force plus and pH force is acid gradient formed, ATP's enzyme that channels hydrogen form the high to low concentration 3 ADPA and NADH 2 ATP and ADH. Source of metabolic energy: substrate level phosphorylation, respiration, chemical reduction of an electron acceptors through a specific series of the electron carriers in the metabolism membrane. The electron acceptor is commonly oxygen, but carbon dioxide, SO4 and nitrous oxide are employed by the some microorganism. Photosynthesis similar to the respiration except the reductant and oxidant are the created by the light energy. Respiration can provide the photosynthetic organism with energy in the absence of the light. The Krebs cycle intermediate the compounds serve as a precursor molecules, building blocks for the energy requiring photosynthesis of the complex organic compounds in bacteria degradation reactions that simultaneously produce the energy and generate the precursor molecule. For the biosynthesis of the new cellular constituents are called the amphibolic. Energy requirement is oxidation of the organic compounds, chemotrophs, sunlight is a phototrophs. Metabolic requirements, carbon source, autotrophs, lithotrophs is used carbon dioxide as a C source. Phosphosynthetic autotrophs use light the energy. Chemolithotrophs use the inorganics. Heterotrophs use organic carbons. Glucose for the growth. Clinical lab classify the bacteria by the carbon source, lactose, and the end product ethanol. Nitrogen source is ammonium is used as the sole end source and the source by the most microorganism. Ammonium chloride be the produced by the nitrogen by the nitrogen fixation or from the reduction of the nitrate and nitric oxide. Physiologic types of the bacteria existence, energy source, oxidation of the organic, sunlight, phototrophs, compounds, chemotrophs, carbon source. Organ is a heterotrophs, inorganic is a autotrophs, electron donor, organic organotrophs, and inorganic is the lithotrophs. The metabolic requirement is sulfur source, a component of the several coenzyme and amino acid. Most microorganisms can use a sulfate as the S source. Phosphorus source, a component of the ATP nucleic acid coenzyme. Phospholipids, thiouic acid, and capsular polysaccharide is also required for the single transduction. Is phosphate is usually used as a P source. Mineral source required for the enzyme function. For the most organism is necessary to provide the source of the potassium, magnesium, calcium, iron, sodium, and chloride. Many other minerals is can be provided is the tap water as a contaminants of the other medium ingredients. Uptake of the ferric acid is facilitated by the production of the siderophores, iron chelating compounds, enterobactin, growth factor, organic compounds, amino acid, sugar, nucleic acid, vitamins is a cell must contain in order to grow, but which is unable to synthesize. Purines and pyrimidine required for the synthesis of the nucleic acid, which is DNA and RNA. Amino acid required for the synthesis of the protein. Vitamins need as a coenzyme and functional group of the certain enzymes. The transport system, the protein that mediate the passage of the solutes through the membrane are referred to as a transport system. Carrier protein, porters, and other transport system is operated by the one of the three transport process in a unipoint process. The solute passes through the membrane unidirectionally. In the symport process, two solute must be transported in the same direction at the same time. In an antiport process, one solute is the transport in the one direction. This simultaneously as a second solute is transported in the opposite direction. You can see the transport system: uniport, semiport, and antiport. Diffusion system: passive diffusion, facilitated diffusion, ion driven transport, binding protein depending transport, and group translocation. Membrane is a selectively permeable. Few molecules passes through the freely movement involve the both active and passive process. The passive process, no energy ATP is required along the gradient. Simple diffusion, facilitative diffusion, osmosis. Simple diffusion, facilitated diffusion can reduce a concentration gradient but cannot create the one. The osmosis is a pressure, osmotic pressure. So in this slide we can see the active process, energy ATP is required, active transport and group translocation. Facilitative diffusion, you can see the extracellular and intracellular. You can see the extracellular, intracellular active transport. This is the transport system, solute, facilitated diffusion, active transport and group translocation. The temperature, one of the most important factor, optimal growth temperature, temperature range at which the highest rate of the reproduction occur, optimal growth temperature for the human pathogens. Microorganism can be categorized based on the optimal temperature requirements like a polychlorophylls, mesophils, which is grow on the 20 to 40 centigrade, thermophils in 40 to 90 centigrades. Most bacteria are the mesophils, especially the pathogens that require the 70 centigrade. Bacterial temperature requirements 150 0 is a variable. So you can see the 0 centigrade as a polysaccharofilic, and second one is mesophilic, and third one is thermophilic, which is grow and reproduce on the 90 degree, and mesophilic is a intermediate which is grow on the 37 centigrade. Effect of the temperature can growth. Temperature, you can see the sum will exist below the 0 centigrade if liquid water is available like oceans, refrigerators, freezers. Pigmented bacteria in Antarctic ice. The mesophiles, most human flora and the pathogens are the mesophiles, thermophiles. Hot springs, the hot springs effluates from the laundromats, deep oceans, thermal vents. The respiration in bacteria, obligate aerobe, microaerophilic, obligate anaerobe, facultative anaerobe. The facultative anaerobes means aerotolerant anaerobe and capnic bacteria. Categories of the oxygen requirement, aerobe utilize oxygen and can detoxify it, obligate aerobe cannot grow without the oxygen and cannot grow without the oxygen is a mycobacterium tuberculosis, micrococcus, bacillus, pseudomonas. Facultative anaerobes utilize the oxygen but can also grow in the absence. The microphilic requires only a small amount of the oxygen, Helicobacter and Lactobacillus. The anaerobe, which categorize of the oxygen requirement, does not utilize the oxygen. Obligate anaerobe lack the enzymes to detoxify the oxygen, so cannot survive in an oxygen environment. Clostridium and bacteroids. The aerotolerance anaerobes do not utilize the oxygen but can survive and grow in its presence. Streptococcus pyogenes. Carbon dioxide requirements. All the microbes require some carbon dioxide in their metabolism. Capanic grow best at higher carbon dioxide tensions than normally present in the atmosphere. You can see the oxygen which is obligate anaerobe. Second one, you can see and evaluate the growing of the bacteria, which is called the facultative anaerobe. And third one, the third slide, you can easily evaluate the obligate anaerobe. The four toxic form of the oxygen is singlet oxygen, superoxide radicals, peroxide anions, and hydroxy radicals are found in the during the phosphosynthetics, and molecule oxygens with the electron are the booster to the higher energy state during incomplete reduction of the oxygen in anaerobic and aerobic respiration. The third one is during reactions that neutralize the superoxide radicals from ionized radiation and from the incomplete reduction of the hydrogen peroxide. The other one are the neutralized by the carotenoids that remove the excess energy of the single oxygen. The superoxide radicals, superoxide dismutase enzyme that detoxify the them, catalase or peroxidase enzyme that detoxify the peroxidase anion, the catalase, peroxidase, and antioxidants such as vitamin C and E that protect against the toxic oxygen production. The enzyme and their role in metabolism. The enzyme organic catalysts of the higher molecule structures are produced by the living cells. They are of a protein nature and strictly specify in action and play an important part in the metabolism of the microorganism. And in the metabolism of the microorganism, they specified and associated with the active centers formed by a group of the amino acids. So some enzymes are excreted by the cell into the environment for the breaking down the complex colloid nutrients materials, while the other enzymes are the contained inside the cell. The bacterial enzymes are the subdivided into the some groups: hydrolase, transferase, oxidase, and isomerase, and lysase and ligases. The hydrolases, which catalyze the breakdown of the link between the carbon and nitrogen atoms, between the oxygen and sulfur atoms, binding one molecule of the water. The transferase perform a catalysis by the transforming the certain radicals from the one molecules to the another molecules. The third one is oxidative enzyme, which catalyze the oxidation reduction process. The isomerase and racemase play an important part of the carbohydrate metabolism. Rearrangement the atom of the molecules. Lyses remove the chemical group from the molecule without adding the water. Like gases join the two molecules together and usually require the energy from the ATPs. You can see the enzymes, which is active enzyme and substrate, and significance of the enzyme with the help of the amylase produced by the mold, fungi, and starch is stratified and this employed in the beer making industries, alcohol production, and the bread making. The proteinases produced by the microbes are the found proteins produced and microbes are the used for the removing their hair from the hide, tanning hides, liquefying the gelatinous layer from the films during the regeneration, and for the dry cleaning. Fibrinolysis produced by the streptococci dissolve the thrombi in human blood vessels. Enzyme which hydrolyzes cellulose aid and either assimilation of the rough folders. Due to application of the microbial enzyme, the medical industries has been able to obtain the alkaloids, polysaccharides, and steroids, Bacteria play an important role in the treatment of the coffee, coca, and tobacco. Enzyme permit some species of the microorganism to assimilate methane, butane, and other hydrocarbons and the synthesize complex organs compounds from them. With the help of the enzymatic ability of the yeast in special type industrial installation, protein vitamins concentration can be obtained from the waste products of the petroleum paraffin. You can see the metabolism result in the reproduction, the microbial growth and increase in the population of the microbes rather than the increase in size of the individuals. Result of the microbial growth is discrete colonies and aggregation of the cell arising from the single parent cell. Reproduction result in the growth. Binary fusion, division exactly in a half, most common means of the bacterial reproduction, forming the two equal size progeny genetically identical offsprings, cell divided into the geometric progressions, doubling the cell numbers. You can see the size and the cells are the doubling. Binary fusions doubling time is a unit of the measurement of the microbial growth. The culture growth. The growth of the culture goes through the four phases with the time, lag phase, log and logarithmic phase, stationary phase, death or the decline phase. You can see the lag phase and then you can see the log phase. Third one is a stationary phase and fourth phase is the death or the decline phase. Lag phase. Organisms are the adjusting to the environment. You can see the little on the node vision synthesize the DNA, ribosomes and the enzymes in order to break down the nutrients to be used to the growth. Logarithmic phase division is at the constant rate generation time. The cells are the most susceptible to inhibitors. The stationary phase dying and dividing the organism are at an equilibrium. Death is due to the reduced nutrients, pH changes, toxic waste and reduced oxygen. Cells are the smaller and have fewer ribosomes. In some cases cells do not die but they are not multiplying. Stationary phase, death phase. You can see the bioreactors, enumeration of the bacteria. For example, the variable plate count. The second one is the direct count. And the third one is the most probable numbers. Viable plate count, most common procedure for the assessing bacteria number, serial dilutions of the suspension of the bacteria and are the plate are incubated. The number of the colonies developing are then counted. It is assumed that each colony arises from an individual bacterial growth cells. The viable plate count. By the counting the colonies and taking into the account the dilution factor, the concentration of the bacteria in original sample can be determined. Only the plate having the between the 30 and 300 colonies are used in the calculation. The viable plate count. The viable plate count 5 multiple number of the colonies time to the diffusions and dilutions factor to find the number of the bacteria in the sample cells. Example, the plate count is 54, dilution factor is 1 the 100 of the 10,000 ml. Calculation, 54 into the 10,000 is a 5,40,000 bacterial per ml. TNTC If the number of the colonies is too great over the 300, the sample is labeled as a T and TC, too numerous to count. Limitation of the viable plate count, selective as to be the bacterial type that will grow the given the incubation temperature and the nutrients types. Viable plate count, dilution factor of the 1 by the 1000 and click to the incubate. So this is, you can see the colonies of the bacteria will grow simultaneously. 417 colonies, T and TC. Dilution factor is 10 lakhs. So you can see the colonies will generate 22 colonies. Too few, the count is less than 30. The viable plate, next you can see, click the incubate. You can see the 42 colonies, means calculate the number of the bacteria per ml. Calculate 42 colonies, dilution factor is a 1 lakh. 42 into the 1 lakh is 42 lakhs bacteria per ml. Nutrients media, ordinary simple media, special media, serum agar, serum broth, coagulative serum, potatoes, blood agars, blood broth. Elective media, enriched media, differential diagnostic media is the proteolytic actions, fermentation of the carbohydrate, hemolytic activity, reductive activity of the microorganisms, media containing the substance assimilated only by certain molecules. Biochemical properties, you can see the colonies, which is a flat, rise, convex, erose, lobate, and entire curd. So you can see also colonies shapes. This is also colonies. Pure culture isolation. You can see the sample incubations, incubator isolations, inspections, identifications, DNA analysis, microscopic examination, the bird embryos, and you can see the special immunologic or the molecule test. So isolated colonies obtaining the steps of the streak. You can see the steps in the loop delusions. So this is our today the presentations. We'll meet you soon.